Namaste, myself Shivi Saxena, Faculty of Physiotherapy and Diagnostics and Discipline of Medical Laboratory Technology. My topic is Balantidium coli. History and distribution. It was first described by Malmenst in 1857 in the feces of dysenteric patients. It is present worldwide, but the prevalence of the infection is very low. The most endemic area is New Guinea, where there is a close association between man and pig. Habitat B. coli resides in the large intestine of man, pigs, and monkeys. Morphology: B. coli occurs in two stages: trophozoite and cyst. Trophozoites the trophozoite lives in the large intestine, feeding on cell debris, bacteria, starch, grain, and other particles. The trophozoite is actively motile and is an invasive stage of the parasite found in dysenteric stool, and it is a large ovoid cell about 60 to 70 μm in length and 40 to 50 μm in breadth. Very large cells measuring up to 200 μm are sometimes seen. The cell is included within a delicate pellicle showing longitudinal station. The motility of trophozoites is due to the presence of short delicate cilia over the entire surface of the body, and its anterior end in narrow and posterior end is broad. At the anterior end there is a groove leading to the mouth and a short funnel-shaped gullet cytopharynx. Posteriorly, there is a small anal pore, cytopes. The cilia around the mouth are large adrenal cilia. The cell has two nuclei, a large kidney-shaped macronucleus, and laying in its concavity a small micronucleus. The cytoplasm has one or two contractile vacuole and several food vacuole. This is a structure of trophozoites. Next, cyst. The cyst is spherical in shape and measures 40 to 60 μm in diameter. It is surrounded by a thick and transparent double layered wall, and the cytoplasm is granule, macronucleus, micronucleus, and vacuole are also present in the cyst. The cyst is an infective stage of B. coli. It is found and chronic cases are and carries. Life cycle of B. coli. B. coli passes its life cycle is in one host only, monogeneous. Natural host pig, accidental host man, reversible pig, monkey, and rat. Infective form cyst. Mode of transmission Balantidium is a zoonosis. Human beings acquire infection by ingestion of food and water contaminated with feces containing the cyst of B. coli, and infection is acquired from pig and other animal um, reversible from human carries. Once the cyst uh, is ingested. Excitation occurs in the small intestine. From each cyst, a single trophozoite is produced, which migrates to large intestine, and liberated trophozoites multiply in the large intestine by transverse binary fission. And sexual union by conjugations also occur in frequently, during which reciprocal in exchange of nuclear material take place between two trophozoites enclosed within a single cyst wall. Incystation occurs as the trophozoite passes down the colon or in the evacuated stool. In this process the cell round up and secretes a tough cyst wall around it. The cyst remains viable in feces for a day or two and many contaminate food and water, thus it is transmitted to other human or animal. Pathogenesis. In a healthy individual, B. coli lives as lumen commercial and is asymptomatic. Clinical feature: most infections are asymptomatic. Symptomatic disease of Balantidiasis resembles amoebiasis, causing diarrhea or frank dysentery with abdominal colic, nausea and vomiting. Balantidium ulcer may be secondarily infected by bacteria. Occasionally, intestinal perforation, peritonitis, and even death may occur. Rarely, there may be involvement of genital and urinary tract. In chronic balantidiasis, patients have diarrhea alternating with constipation. Laboratory diagnosis. Um, Stool examination. Diagnosis of B. coli infection is established by demonstration of trophozoites and cysts in feces. Motile trophozoites occurs in feces, and cysts are found in formed stools. The trophozoite can be easily recognized by their large size, macronucleus, and rapid involving motility. And cysts can also be recognized in the formed stools by their round shape and presence of large macronucleus. And biopsy: when stool examination is negative, biopsy specimen and scrapings from intestinal ulcer can be examined for presence of trophozoites and cysts. And culture: B. coli can also be cultured in vitro in lox egg albumin medium or NH polygenic medium like Entamoeba histolytica, but it is really necessary. And treatment: tetracycline is the drug of choice and is given 500 mg 4 times daily for 10 days. Alternatively, doxycycline can be given. Metronidazole and nitronidazole have also been reported to be useful in some cases. Thanks.